This is the lecture. Remember I mentioned that there was a lecture that was cut out, and I don't know if you got the new schedule, but today was scheduled for hypersensitivity. This entire class was cut out of the lecture. I have no idea why. No idea at all, but it was put back in because I was broke up. Just to put things in perspective, what we've built over the last month with me is that there are two responses, innate and adaptive. The innate is the older response, evolutionarily older. It does not have memory, it does not have specificity, it does not have diversity, which we talked about yesterday. We didn't talk about memory, we talked about specificity and diversity yesterday, and how you got there. If the innate, and we talked about how cells, what the cells' innate response are, how the cells' innate system respond, how they recognize pathogens, and I mentioned briefly that there's got to be a link between the innate system and the adaptive system, and that's what we're going to cover today. So we're covering, one, the link between the two. If the innate fails, what happens next? And then we're going to talk about how the adaptive system gets started and how it is regulated. So the four topics behind me, remember them all, are closely interchanged. Major histocompatibility is actually a series of proteins that are involved in the process of antigen presentation and processing. Actually, that's actually in the wrong order. So processing occurs first and presentation second. So the top three topics are somewhat interrelated, and you'll appreciate that in the next half hour. And then T-cell activation is the antigen-presenting cell, the cell that is in the big toe, that is collecting fragments of the pathogen. Then migrates to the draining lymph node, where it seeks to communicate with a T-cell. And the T-cell it's going to communicate with is a CD4 positive T-cell. I hope a T-cell I mentioned to you a couple of months ago when we first met, that you just have to memorize helper, CD4 helper, and just keep memorizing that, and I'll put it on the exam, and pretty much just that way. The helper T-cell is going to communicate with this antigen presentation process. It's going to become activated, a variety of events will occur, and then that helper T-cell will help the immune system. And it will guide the response, an appropriate response for the type of pathogen that was encountered. And at that point, we've gone full circle. And then everything that remains for you to learn is going to be the immunopathology associated with the system failing. Okay, so let's get started. So there are three antigen-presenting cells. Let me change what I just said. There are three what are called professional antigen-presenting cells. Professional meaning that is a major responsibility. You'll notice on the right-hand side there are a bunch of cells listed that also can present antigens. They're not professional. It means they have the capacity to do so, but is not their main cell function. So the three on the left are the ones we'll talk about in the next slide. Dendritic cells are found in tissue. So they're present in tissue and in blood, but they're present in tissue, and if indeed the innate system fails, they will process and present antigen. Macrophages, we know macrophages are differentiation products of monocytes, which are brought into the cell, into the tissue, after neutrophils. And then B cells we talked about yesterday. We're going to actually discuss B cells per se. The B cells are born in the bone marrow. When they're born, they make IgM, and they circulate around the body and are brought into sites of inflammation. Yeah.
Just a question on that chart. So is it if the macrophage fails, then the glial cell will come in? Like, are they?
No, there's no pecking order.
Okay.
Actually, if there is a pecking order, there's one cell whose only job is antigen presentation, and that's the dendritic cell on the left. Macrophages are still phagocytic and they can still be part of the innate response. And B cells obviously make antibodies as their main function. So while these cells are designed to present antigen, the only one that is slowly responsible, its only responsibility is antigen presentation. These cells need to get angry in order to present antigen, and that stimulus for anger in these two cases are PAMPs. Remind you, PAMPs was an acronym that the main word in that word PAMP, you don't have to memorize what the acronym means. The key word there is patterns. Remember we talked about you can recognize a car, you can recognize a table, versus recognizing a very discrete molecular signature within the car, or within a table or a chair or whatever. So patterns are the broad sweep of something that a pathogen expresses, and you'll notice patterns associated with a pathogen are what bring a dendritic cell from a resting state to an activated state where it can present antigen. Again with the macrophage. A pattern causes it to be activated and therefore very phagocytic as well as present antigen. The B cell is a little bit different because remember, for those of you who made it yesterday and appreciate it, for those who did come to class yesterday, I really do appreciate it. You had an exam at 3 o'clock and that was an extra challenge. So it doesn't need for 2 hours. So I appreciate you showing up. The B cell retains the antibody on its cell surface, and that's called the B cell receptor. Everyone comfortable with what a receptor does? A receptor in a cell has two functions. It has a function facing outside the cell to receive information, to know that the ligand is out there, and therefore the cell must respond. And then it has an intracellular portion that communicates with the cell to get the cell to respond accordingly or appropriately. So the B cell receptor already, we don't need a pattern recognition receptor on B cells because it already has a pattern recognition. It's the antibody, the B cell receptor itself. So those are the three triggers to activate these cells, and the activation process. The activation may be more complex than just antigen presentation, but for today, they're being activated so they can present antigen. You ready on this slide? For your reading pleasure, and also for your studying, many of my slides are repeats. You might be able to study better off of one slide or another, so I've given you repeats. There's nothing on this slide that I haven't really already covered, so you can study off either one of these slides. Okay, this is a complicated slide. We're going to come back to it 4 or 5 times, but I just want you to appreciate what antigen presentation means. And the antigen presentation means, so here is a molecule, this red molecule right here. What's the cluster? Oh, this cluster, no. And the blue molecule here, you'll notice, I can't stretch that far. This red molecule here has a little red dot. This blue molecule here has a little green dot. These are antigen presentation proteins. I'll define them in more detail scientifically in a moment. And the red dot or the green dot in each figure is a product of the pathogen. So this antigen-presenting cell, right here, is using the red heterodimer or the blue heterodimer here to present, to expose, to share with another cell something from the pathogen. That's what antigen presentation is all about. The antigen-presenting cell is in the big toe. It takes in chemicals from the pathogen, displays them on these red or blue molecules, and then drains to the lymph node where it will communicate with that CD4 positive T cell. We'll get to CD8 a little bit later, about an hour and a half from now. Everyone good with that? So I haven't introduced you what these proteins are yet. It's written here, but I haven't introduced them yet. And the objective here is to share with that CD4 T cell information about the pathogen, so that CD4 T cell can make a decision. And this is the beginning of why the adaptive response takes 10 days. One, the dendritic cell has to process, or the antigen-presenting cell, as I said, mostly the dendritic cell, have to process the antigen. Then they have to leave the site of damage or injury or infection, drain to the lymph node, and then they need to find the correct T cell. And we haven't talked about that in detail, but we will in a moment. That not every T cell coming through the lymph node can interact with this antigen from a given pathogen. There's only going to be one T cell in the entire body. So this antigen-presenting cell can be sitting in the lymph node for days until the correct T cell comes along. Then, when the T cell and the antigen-presenting cell communicate, that's another day where the information is being shared. Then the T cell has to respond, and that's still 2 or 3 days. Hence the 10 days of an adaptive immune response. Those are the reasons for the delay, if you want to call it a delay. You could call it a delay, but those are the reasons for the time frame in which it takes to generate an adaptive response. I think I'll do it here. I mentioned that when the antigen-presenting cell does ultimately find the one T cell that can respond to this one antigen, they're going to spend a day together. Two cells in communications, adherent to each other, aren't just going to adhere. It's very unlikely that just the interaction shown right here is enough to allow these cells to adhere for an entire day. So this entire complex, it's now been given the name of a synapse. So if you think about neurobiology, when two cells come together and there's an exchange of neurotransmitters, that's the synapse. The immunologists stole that word and they talk about this synapse. There are a number of molecules that are involved in information exchange that's going on during that one day, and there are a number of molecules involved in adhesion so the cells don't separate. So this is a very complicated organelle, if you will, of the cell that enables these cells to be in communication for 24 hours or so and provides information exchange. Okay. We have two types of antigen-presenting proteins. I haven't given them a name yet, but notice that one is called class 1. You saw in the previous slide, you may have noticed. There was a class 1 and class 2 as well. I didn't point that out on purpose. There's a class 1 molecule and a class 2 molecule for presenting antigen. The class 1 molecule only communicates with CD8 positive T cells. You just have to memorize that. There is no trick I can give you to explain the relationship. So class 1 molecules present antigen to CD8 positive T cells. Class 2 molecules on the right-hand side present antigen to CD4 positive T cells. So 6 things you have to memorize. CD4 is helper. Helper is class 2. CD8 is cytotoxic. Cytotoxic is class 1. Those 6 pieces of information you just have to memorize. I have no other way of knowing how to teach it to you. So I'll say it again. CD4 are helper T cells. They communicate via a class 2 antigen presentation. CD8 T cells are cytotoxic T cells, communicate via a class 1. So those 6 pieces of information just memorized. They will be on the exam, and I can't explain them except that's the way it works. The structure of the antigen-presenting molecule, which is this purple, pale blue, greenish, and red. It's a little different between class 1 and class 2, but look at the structure. Take a step back and don't worry about who's connected to whom. Notice that structurally there are 4 globular components that make up this molecule, 4 globular components that make up this molecule, and the groove, the site at which this foreign antigen sits in the MHC for it to be presented. It really is a groove, and it really does look like this laser pointer. There is a groove. There's a groove right here and a groove right here, and it really does look like a groove, and the peptide, in most cases, I mentioned this yesterday when we talked about antibodies, that even though antibodies can bind to every chemical signature out there, including signatures that are on Mars that we haven't seen yet. In general, antibodies bind to proteins. That's the vast majority of the antigen for an antibody. The vast majority of the antigen that fits in this groove is also a protein. So that's why it's called a peptide binding groove, because there are other things that can fit in the groove, but in general it's a peptide, not in general. Majority. So it truly is a groove. It has walls. My fingers are the walls, and then it has a platform, which is my palm, and the peptide truly sits in that groove. I think I'll have 1 or 2 cartoons to show that a little bit better than I can do with the pointer. So even though you'll notice that this is one protein, this is the second protein. Here, this is one protein, and the red is the second protein. Doesn't matter, that's detail you don't need to worry about. The message is that it's a 4 globular shaped molecule. It gets there different ways, but it accomplishes the same thing. It creates this groove right here. Yeah.
So is the groove where you're presenting the antigen?
Yes. The peptide antigen will be sitting in that groove up here, communicating with the T cell. So this is the same picture. This is the same picture as the previous one. Here's the groove, here's the peptide, and it's going to be communicating with a molecule on the T cell. That is the presentation process. So here's a ribbon diagram of the walls of the groove. There are beta sheets that form the floor of the groove. There are alpha helices that form the wall. And this is the structure for class 1. Class 1 is what T cell? CD8. So here is what the groove looks like for class 1. And here's what the groove looks like for class 2. Class 2 is CD4. Class 2, CD4, for CD4, class 2, helper. Helper, CD4, class 2. And they look the same. Yeah, are there subtle differences? Of course. But we've got alpha helices forming the wall, and the beta sheets forming the floor of the groove. Now, I've placed a peptide into the groove. So the orange is a peptide from a pathogen. And you can see now that if you're a T cell, if you out here are the T cell, so the antigen presenting cell is in the wall of this room and into the men's room. Men's room out there? Whatever's back there. And this is the surface of the antigen presenting cell, and we are the T cell. So we're looking at the antigen presenting cell, and look what we see. We've got this nice orange peptide staring at us. That's how the T cell knows. That's how antigen, using our expression, antigen presentation, that's how antigen has been presented. Because the cell that's in the hallway out there is exposing this molecular structure to us, who are the T cells looking down on it. There is a little difference, nothing you should worry about, but it ends up that the peptides for class, peptides for class 1 are smaller. So this is closed off here and here, so a peptide has to fit in the groove. So it's not a hot dog. So this is a hot dog bun where the hot dog can be bigger than the bun for class 2. This for class 1, the hot dog has to fit into the bun. It can't be a bigger hot dog. So just a minor difference between the structures, no big deal. I do not have a biological explanation for why one or the other. It ends up that the molecule that is doing this presentation is called a major histocompatibility protein. MHC, I think, was on the first slide, or maybe I said major histocompatibility. No, I actually called it major histocompatibility during the first slide. It's a major histocompatibility protein. So now we need to take a side road for a couple of minutes. Histocompatibility. Mean anything to you? Does something pop into your mind when you hear major histocompatibility? Does some biological or clinical concept pop into your mind at all with just those words? I want to project the words so you can stare at them a minute. I don't know if I'm going to have to go back to the first slide. Oop. That's MHC, it doesn't have the full. Let me go back to the first slide so you can stare at the word. So major histocompatibility. Anything come to mind? Does it get any feel for that term? Okay, let's take it apart. Oh, yeah. Excuse me? Graft versus host. Transplant. Histo, tissue. Compatibility. Compatibility is an English word, not a scientific word. Tissue that is compatible, meaning if my liver goes into one of you, you're either going to reject it or not reject it. Is my tissue acceptable to your immune system? You know about transplant, you know about transplant rejection. You know about the number of drugs the people who receive transplants, immunosuppressive drugs, the people who receive transplants must take. So why? Why? I'm human, and sure, I'm maybe a little bit, I'm a man, she's a woman. My genetics are different from someone from the Middle East or someone from Africa or South America or whatever. But within some bigger domain, we're humans. So why would a protein, or why would a tissue from me be rejected by most of you? It ends up that I express MHC molecules, major histocompatibility molecules, so I express class 1 molecules and class 2 molecules, as do everyone in this room. We all express class 1 and class 2 molecules. And they are called, while their job is antigen presentation, they were found because they are the source of tissue incompatibility. So for some reason, the gene sequence for my class 1 molecule, MHC class 1 molecule, antigen presentation of the class 1 type, my protein sequences for MHC molecules of the class 2 type, for the CD4 cell. The sequence of my proteins is different from most likely everyone in this room, and so on around the world. And therefore, when my tissue goes as a transplant to any of you, my protein sequence for class 1, MHC class 1, my protein sequence for MHC class 1 is different from yours. Your immune system says, wait a minute, I've never seen that stretch of amino acids before. It's foreign. I'm going to reject it. Because that's my job as an immune response. My job is to kill and eliminate anyone whose sequence isn't me. So my tissue transplant for you guys will be rejected because the sequence of my class 1 and class 2 molecules are different. That's how the name came about, but that's not their function. They don't exist so that we get transplant data. They exist because there's a great deal of heterogeneity in the genetics of class 1 and class 2 proteins. Why? Why would the gene sequence or the protein structure for my class 1 molecule be different from... Why is the class 1 molecule? There are 30 people in this room right now. There are 30 different class 1s. Actually, there are 60. Actually, there are 90. We'll get to that. No, there are 120. We'll get to that. So let's make it simple. Class 1 molecule, 1 gene. Why do we have 30 different gene sequences for class 1 molecule? And the class 2 molecule as well.
If the whole system is based around seeing something foreign and responding to it in all the ways that we've been talking about, it would seem it's foundational that you have to know what not to kill. And
What not to
kill.
Not to kill. Okay, so that's going to be phagocytation. And
so if you have, you need some way to recognize these are the things that are supposed to be here. That's the only way you can recognize what's not supposed to be.
Okay, all that you said is absolutely true, and all that's happening on the T cell side. The T cell is making that decision about whether something should or should not be there.
Is it not based on the MHC presentation of the cells then?
Right, now we're getting closer, we'll bring it back to presentation. Remember, MAC, let me bring the picture of the peptide again. That might help this conversation.
If you're trying to build a system that's extraordinarily sensitive, then each individual phenotype walking around would have to be uniquely sensitive and
You're getting there, you're getting much
forming
more.
the system.
Remember, these peptides have to fit in the groove. The groove's gonna have a shape, both a shape and a chemical, the side chains. The groove is made up of amino acids. The alpha helices are on the wall, the floor is the beta sheath, but the side chains of the amino acids are gonna be pointing into the groove. So not every peptide can fit into every groove. Does that make sense, everyone? The groove has a chemical personality. And therefore the peptide has to be able to fit into the groove, the chemistry. If the groove has a whole bunch of negative charges facing, if the wall of the groove and the floor of the groove happen to have amino acids where a lot of negative charges are facing into the groove, a peptide with a lot of negative charges is not going to be able to fit into that groove. That's chemically incompatible. Okay, so not every peptide can fit into every groove. That should all be comfortable with that. That was a hint. Why do we have so much heterogeneity in the genes for MHC class 1 and class 2, given what I just said, that not every peptide can fit into every group?
To expand the diversity of the antigens?
Yeah, exactly. If I get hit with Ebola and my group is incompatible with all the peptides from Ebola, I'll die. But we got 30 different grooves in this room. One of your grooves will allow the Ebola peptide to fit in, and your immune system will respond, and you'll live. So from a species perspective, the breadth of MHC that's going on in this room and in this world is, from a population perspective, some peptide, some groove somewhere will fit. Some groove in the 3 billion people now on Earth? I think we're 3 to 4 billion people. Some peptide, some groove in 3 to 4 billion people, hopefully one man and one woman, will fit the peptide of that pathogen, and then that pathogen, that peptide can be presented, as shown in orange here. The T cells will respond, that individual, or that cluster of individuals, will survive. If we all have the exact same groove, meaning the exact same MHC molecules, we're going to be limited by what can fit. And if a pathogen comes up with peptides that can't fit, that pathogen wins and we lose. But if we have an enormous amount of heterogeneity in the all population, we'll cover every single peptide possible. And we got that? Comfortable with it? Now that's why we reject organs. That's why my organ is incompatible with everyone in this room, because my peptide group is different in sequence from all your groups. So the name has nothing to do with function, but okay. We've had that peptide sitting in the groove. There are hot spots. There are certain amino acids in red here whose, because of the way the beta sheet is folded, because of the way the alpha helices are folded, their side chains are facing into the groove. So those red hot spots have side chains facing into the groove. So those hot spots are determining what peptide can fit and what peptide can't fit for any given group. Yeah, this is I mentioned that the class 1 has walls on the side, so it can only fit 8 to 10. The class 2 is more open-ended, so we can fit the hot dog with the hot dog hanging out on the outside. Names, don't worry about the names of the peptides. Ends up the good of the MHC. Ends up the good news is peptides within the groove can flex a little. So a peptide that aren't the whatever color this is. Yellow peptide is fitting right in the groove. But the orange peptide can make or the red peptide can create a little bulge, and therefore the groove is not, the groove is not so strict in its binding capabilities of peptides that only one peptide can fit, you know. If luckily, so if the yellow peptide goes 1, 2, 3, 4, 5, 6, 7, 8, 9, 4 and 5 have to be compatible with the group. But 4 and 5 can also bulge out, and therefore a peptide whose 1, 3, 6, 7, 8 fit in the groove will fit just as well as 1 through 9 all fit. So there's some flexing going on. We use the word flex when we talked about C3 and complement. There's some breathing that can go on, and different peptides can find the side chains and allow them, these side chains, these hot spots in red, different peptides are compatible with many different amino acid sequences, are compatible with fitting into this group. Are we okay? Because now we have to name these guys. I mentioned that there are 30 different MAC class 1s in this room, and then I raised it to 60, then 120. I'm going to now explain why that number can grow. Yeah.
For cases where an organ transplant is not rejected, is that just because it's compatible enough?
Yeah,
It's
exactly.
not-
So tissue transplant, why, you know, every time there's a call on the radio, do you have bone marrow that's compatible with, you know, let's do a test. When people are doing tests before a transplant, they're asking, what is the MAC sequence? Now we're at a point of sequencing. In the old days, it used to be antibodies and so on, but we've gotten more and more sophisticated. So, you know, what is the MAC on being expressed by the donor, and what is the MAC being expressed by the recipient? And if they're very, very similar, if they're identical twins, they're identical. But if they're very, very similar, that tissue is less likely to be rejected. But if I have genes 1 through 6, and you have genes 10 through 12, 10 through 15, there's no way there's going to be any compatibility in them. So that's what tissue typing is all about. Okay. Don't worry about the mouse. Well, these two you guys are not mouse biologists. Just worry about human. I should have wiped that out. There are genes, more than one gene for class 1 and more than one gene for class 2. There are actually 3 genes for class 2, 3 genes for class 1. It works out really easy. If there's only one letter describing a gene, it's class 1. So if I say MACA, A is the only letter that I said, it's got to be a class 1 gene. If I say MACDR, it's got 2 letters, and means it's a class 2. So the number of letters describing the gene puts it into class 1 and class 2. Don't worry about class 3. So it ends up, we have 3 genes that code for a class 2 molecule. We have 3 genes that code for a class 1 molecule. That's why that 30 became 60 and started to grow. It's even more than that. You got 3 genes from mom for class 1. You got 3 genes from dad for class 2. They probably are not the same genes. So now you have 6 class 2 genes. You also have 6, 3 from mom, 3 from dad, class 1 gene. That helps with keeping you alive. So you don't have one MAC with one groove to keep you alive. You now have 6 MACs to keep you alive. And now, therefore, in this room, 30 times 6 is we've got 180 different MAC grooves in this room. Hence, at some point, we get to a point where every peptide in the world will find a groove.
So would you say that you have 12?
And this is the mom and dad give you, you know, mom has A and D, you have dad, that's dad. Dad is A, okay. Dad is A and D, mom has C and D, therefore you have AC, you have every combination. So you have all from mom and all from dad. That's why also siblings can or cannot be good matches for tissue transplant. Yeah.
So would you say that you have 12, since you have 6 class 1 and 6 class 2 and those are not the same, so overall you would have 12 NHGs?
Yeah, but class 1 and class 2, class 1 talks to CD8, class 2 talks to CD4, so if you need a CD4 response, you only have 6 to help you. If you need a CD8 response, you only have 6 to help you. The class 1s can't, if you need a CD4, those class 1s are doing nothing. So I'd rather you think about 6 rather than 12. All right, now, how many genes are out there? What kind of diversity are we talking about? And these are the numbers. Remember, HLA, I haven't introduced it, but I guess I have to now because of this slide. MHC and HLA can be used interchangeably. HLA stands for human leukocyte antigen. You don't have to write that down. But someone might talk about HLA-A, or they might call it MHC-A. Okay. Notice, well, I don't like this slide, but notice that HLA-A, B, E, G, there's one letter, that means they're all class 1s. Here, it's actually 2 letters, don't worry about the B's and the A's, that's, they made, there's 2 letters. It's either DQ, DR, or DP. 2 letters, class 2, 1 letter, class 1. And notice what kind of diversity we're talking about. And that's why tissue typing becomes so difficult. Because we've got more than 1,000 different HLA-As, genes, out there in the human population. Okay, that's presentation. Now we have to get the peptide onto the MHC molecule. That's processing. The presentation is dropping the peptide into the groove and displaying it to a T cell, the appropriate T cell, class 1 CD8, class 2 CD4. Now processing is getting them out, the peptide into the groove. One more thing you have to remember. Memorize. And yeah, do memorize it because I'll put it on the exam. Class 1 molecules work with endogenous antigens. Gotta memorize it. Can't explain it any other way. Well, I can, but it's complicated now. It's probably easy to memorize. Class 2 molecules get their peptides from exogenous route. So the peptide that loads into a class 2 molecule comes from outside the cell. The peptide that loads into a class 1 molecule is within the cell. Endogenous within, class 1, exogenous outside, class 2. So the source of the peptide can either be in the cell or outside the cell. If it's outside the cell, it loads as class 2. So let's do class 2 first, it's easier. Class 2 is, here's the exogenous peptide. Here's the whole antigen. Remember these peptides, you may have noticed, I didn't emphasize it, the peptides are 8 to 13 amino acids long. Most proteins are 100 to 1,000 amino acids long. So these are little peptide chips of a very large protein. So the antigen, the bacteria, the fungus, the parasite, the virus, not the virus, the parasite, the fungus, and the bacteria, which live outside cells. Remember, outside, exogenous class 2, bacteria, parasites, and fungi, almost always, but not, there are exceptions, never enter the human cell. So their proteins, their antigens are going to always be outside. Therefore, they're going to be loading the class 2 molecule, loading onto a class 2 molecule. So the entire protein is large. It's going to be endocytosed. Remember, we've got pinocytosis, endocytosis, and phagocytosis. There's a lot of phagocytosis going on in inflammatory sites. Macrophages phagocytose, they're antigen-presenting cells. There's a mechanism by which we can bring things from outside into the cell. Dendritic cells endocytose. They're not really phagocytic, and therefore when they pull up a little bit of liquid, they're going to get some exogenous peptide, bacterial parasitic or fungal, and being brought inside the cell. The process of phagocytosis or endocytosis puts the antigen in a vesicle. It's not in the cytosol, it's still, topologically, it's still outside the cell. And if you want to talk topology, we can do it during the break. We'll do it offline. But topologically, because this plasma membrane seals right here, the peptide is in the lumen of the endosome, which puts it still outside the cell. Well, coming in from when we synthesize MHC class II molecules are proteins. They're made on ribosomes. Ribosomes put proteins that are going to be excreted or put on the cell surface into endosomes like this. And here we have an endosome expressing a class II molecule. Here we have an endosome expressing the exogenous antigen. These endosomes have lytic proteins, proteases in there. So this antigen is going to be cleaved into peptides. Then this endosome and this endosome fuse, and bingo. You have the opportunity for the peptide to fit into the groove if it can fit. Remember, this protein of 500 amino acids is going to make 490 peptides. 10, 10, 10, 10 through the 500. So there's 490 peptides from just this one protein from the one bacteria. And the bacteria has 10,000 proteins on its surface. So the numbers are in our favor, even though I painted a bleak picture a little while ago, the numbers are in our favor. Yeah. 10,000 proteins on a bacteria, 500 peptides from one of those proteins. One of those 500 peptides times 10,000, so that's 500,000. 500,000 peptides are going to be generated. One of those 500,000 have to fit in the groove. Once it's in the groove, this molecule is placed back on the surface. And remember that picture where I had the T cell and the antigen-presenting cell communicating with each other for a day? Here's the MHC2 molecule. Here's the peptide right in there. It's hard to see in at this resolution. And now this molecule can present antigen to a T cell. This complex can present antigen to a T cell. Everyone got that? Because this one's a little easier than class 1. I'm not telling you the names of the molecules involved because I don't care. Well, you shouldn't care. There probably will be a slide listing the names coming up. The textbook probably has the names, ignore them. They're not important for us today. Everyone good with the exogenous pathway? Okay, now let's switch to the endogenous pathway. Why would a pathogen be inside the cytosol? The virus, exactly. So in most cases, viral proteins are going to be loaded onto class what? 1 or 2? Viruses get inside the protein. Viruses are endogenous. Virus proteins will be in the cytosol. Therefore, they rely on the endogenous pathway. Therefore, they're going to load onto class 1. And class 1 communicates with what cell? T cell? CD8 T cell. What is the function of a CD8 T cell? Cytotoxic. If you have a virally infected cell, what do you want to do to survive? You want to kill that cell. So the virus is using that cell as a home, as a refrigerator. If you destroy the refrigerator, the virus will die. So the whole system works. Viruses load onto class 1. Class 1 talks to CD8. CD8 kills cells. Bingo, the virus is killed when the cell is killed. So it all fits, hopefully fits together for you conceptually so you can help you memorize it. So there's a virus inside the cell. The virus gets broken up. Proteins from the virus are cleaved into peptides. This is the molecule that does it, or the complex that does it. Don't write it down. But again, just like we're doing here, viral proteins are going to be cleaved into peptides. Now we have a little problem. The peptides are where right now? When the virus is sitting in the cytosol, where are the peptides? They're in the cytosol. The MHC molecule, class 1, is ultimately going to have to be put on the cell surface, which means the MHC1, the groove of MHC1, is actually in an endosome. facing into the lumen of the endosome. Because remember, topologically, the lumen of an endosome is outside the cell. So we have to pump these peptides into endosomes. So there's a whole bunch of proteins. They may be on the next slide, listed on the next slide. I don't remember the one. There's a whole molecular system designed to take the peptides from the virus that are in the cytosol, pump them into an endosome, then that peptide can load onto class 1, and ultimately class 1 will present that peptide. Any questions? Mechanistically, I'm not going to test you, and I don't expect you to remember, but you need to be comfortable with topology here, so that you've got who's talking to whom when. I'm more interested in that you're comfortable with where things are and where they're going and why they're going there, and don't worry about the names. All right, what do we got? Yeah, this is the same slide, 10 times more complex. Okay, we're going to do the second also. This is the exact same slide, but now the names are on there. Don't worry about it. Awesome.
I was just curious if like a cell's not infected with the virus, will it just display an endogenous peptide that it expresses?
Good. So, in a healthy individual, MHC molecules are never empty. The groove is always filled with a peptide. In a healthy individual, the peptide being displayed is a self-peptide. That's back to your question. When that antigen-presenting cell with that self-peptide ends up in a lymph node, there better not be a T cell that recognizes it, or otherwise you get autoimmunity. And you'll hear about T cells that recognize self. Hopefully you'll hear about it from someone else. If you don't hear about it from someone else, I'll get that information to you somehow. But yes, that's a very important point. The groove is never empty. And therefore, in healthy individuals, that groove is filled with self.
So then does it,
Stupid design, in my opinion. Really. But that's the design.
if the groove is filled with the self-peptide, then do you need to remove that before the?
No, the peptides, no. It's newly loaded MHC. Once you get an infection, you load new MHC, you don't displace the peptides that are there. So we're constantly making new MHC. We're constantly loading endogenous self-peptides on there until suddenly you have an infection, and now we load the pathogenic peptides. Good question. Just to remind you, the antigen-presenting cells, the message here is that the dendritic cell in a healthy individual doesn't express MHC class 2. We'll now break out class 1 and class 2 biologically. But let's just worry about class 2. The resting cell does not express class 2. When it's activated to a PAMP, meaning some pattern from the bug, it expresses class 2. The macrophage doesn't express class 2. When it's activated by a cytokine or a PAMP, it expresses class 2. Same with the B cell. Well, it expresses more class 2. Little wrinkle, not a big deal. Class 1 is expressed by every cell in the body. Class 2 is only expressed by these cells that are going to the lymph node to talk to a CD4. So we now need to break out a little bit between CD4s and CD8s. So we appreciate what's going on here. Why would every cell in the body express class 1?
In case it's infected,
Right.
they need to see correct.
We decided that class 1 will only load peptides from the cytosol. The only pathogen that really, my 5% rule, the only pathogen that ever enters a host cell is a virus. So a virus can infect, not any given virus, there must be a virus out there that will infect every cell type in the body. Not a virologist. It should be. No. I mean, I have virology going on in my lab, but I have no idea what they do. The virology part, immunology I missed that. So there's a virus that can infect every cell in the body. So every cell in the body might have a virus in it, and we want to kill that factory. Viruses use host cells as factories to make baby viruses. So we want to kill the cell that's virally infected. Therefore, we have class 1 expressed in every cell in the body, because every cell in the body needs to be killed if it's virally infected. Did I lose anyone there? I think I went in a circle, but I hope I didn't lose anyone. Did I lose anyone? What I just said? We're good? Excellent, thank you. Class 2 only taught, and once you're virally infected, you express the viral peptide on class 1. You scream out, kill me, kill me, kill me. The cell that's going to kill you is a CD8 cell. Hence, class 1 CD8. CD8s are cytotoxic, i.e. killing. They interact with class 1, which is on every cell. Kill me, kill me, kill me. The CD8 has the capability of killing a whole cell. So it all works. CD4s are present really only in the lymph node because they're the brains of the operation. Remember I said that there's antigen presentation. You go to the draining lymph node, the antigen presenting cell waits for the T cell, they find each other, they chat for a day or two. The T cell takes that information, decides who do I need to protect my host. That CD4 T cell makes a bunch of decisions, spreads out cytokines to the worker bees, and those worker bees go out and keep you alive. So CD4 cells really only function in the lymph node, and therefore you only need cells draining into the lymph node to express class 2, which are these 3 cells. So we don't need class 2 on a fibroblast. What's this fibroblast in your skin is never going to make it to the lymph node over here. Kind of. Remember this 95.5% rule. So we're only going to drain with these 3 cells, and the CD4s are only in lymph nodes waiting to find their cognate peptide. Hence, professional antigen presenting cells express class 2, all cells express class 1. Do you have that concept? Because that's probably the most important message for the first hour. Anyone uncomfortable with that message? And of course, I'll hang around with the class to clarify. All right. This is a repeat slide. I don't remember why. I'll put it in there. Okay. To talk about the T cell. Why don't we do the T cell in the second hour? Let's take a break. Inside that PRR, remember patterns, pattern recognition receptor. The antigen presenting cell has to get angry. Here's class 2, and here's the peptide in class 2. Antigen presenting cell with that peptide, either the self-peptide or the pathogenic peptide, goes to the lymph node, and T cells are going to float into and out of lymph nodes. T cells are in constant circulation, going in and out of lymph nodes. T cells are unique, similar to what we talked about antibodies yesterday. We talked that B cell 1 makes antibody 1. B cell 2 makes antibody 2. B cell 3 makes antibody 3, and so on. And you have 10 to the 11 different antibodies. It's in circulation right now. You have 10 to the 11 different B cells, coding for each one of those antibodies in circulation right now. T cells, with all the gene rearrangement we talked about, and the BDJ for heavy chain, and BJ for light chain, and the randomness, and the way we took multiplied numbers together more and more and more to get up to 10 to the 11th. All of that DNA craziness that I said is only going on in the B cell, I lied yesterday. It's also going on in the T cell. And the B cell receptor is the antibody stuck in the surface. The T cell has a T cell receptor that has VDJ and VJ and rearrangement and mistakes and exonuclear cleavage and random nucleotides added, all the randomness that I emphasized as much as I could yesterday to get to 10 to the 11th. All of that is occurring in the T cell also. It's not occurring in the heavy chain and the light chain Remember, all that craziness is occurring in the heavy chain and the light chain in the B cell. In the T cell, it's occurring in a chain called alpha and beta. But the gene locus in the T cell that does all that craziness from yesterday is doing all that craziness in this alpha and beta chain. Oh, here's the T cell, sorry. There's an alpha and beta chain of the T cell receptor, just like the heavy and light chain of an antibody. It goes through all this craziness to create a pocket that is unique from T cell to T cell to T cell. That's where the specificity of the adaptive immune system comes in. You have 10 to the 11 T cells in circulation right now. Each of those T cells expresses a unique T cell receptor. Okay? And we get to that unique T cell receptor through the craziness that we talked about yesterday for B cell. That's why the immune system, the adaptive immune system is so specific. Because the T cell that recognizes the peptide that came from Ebola, you only have one. You only have one T cell in circulation right now that recognizes a particular Ebola peptide. Now don't worry, I talk about one, but from a public health perspective, remember, Ebola is going to make 500,000 peptides. So even though I talk about the Ebola peptide in that one T cell is going to keep you alive, remember Ebola has made 500,000 peptides, and you've got potentially a T cell for many of those 500,000. So don't panic. So one T cell or one T cell receptor, and it can only recognize one peptide. And here's that recognition of that. The two chains of the T cell receptor, just like heavy and light chain for antibody, recognize, and this is another transplant issue in a moment, that one T cell can only recognize this peptide sitting in this MHC. So if that peptide sits, let's say you got MHC A14 from mom, and you got MHC gene A99 from dad. The peptide, and just by chance, the peptide can fit into both 14 and 99. This T cell receptor cannot recognize, it is recognizing the peptide and the MHC. So if this T cell receptor is designed random, it's not designed. Remember I mentioned yesterday that you have antibodies against Martian proteins right now, even though you've never been to Mars and never will be. You have T cell receptors that can see every peptide and every pathogen in the world and pathogens that don't exist yet. But that T cell can see that peptide in 14 but not in 99. Or in 99 but not in 14. So the degree of specificity is not just for the peptide, but it's for the MHC molecule that's presenting that peptide as well. So the ligand, so going back to the word receptor, remember receptors have two components. Here's the cell membrane. Receptors have a job outside the cell to grab things, to know that someone's talking to you. And then they have a job inside the cell to communicate with the nucleus to make things happen. The thing a receptor grabs is called a ligand. So using that definition, so a receptor, here's a ligand, a receptor binds to the ligand. Because that happens, things happen inside the cell. The ligand for the T cell receptor is this 3-molecular complex. It's the alpha chain and the beta chain of MHC class 1 and the peptide that fits in the group. That T cell receptor can see only those 3 molecules and no other. That's where the specificity of the adaptive immune, just like we talked about antibodies having incredible specificity yesterday, the T cell receptor, because it uses the same molecular genetic tricks to get 10 to the 11 copies, 10 to the 11 distinct T cell receptors, its specificity is as great as for an antibody. Okay. I am going to mention autoimmunity for just a moment. The classmate brought it up earlier in the hour, last hour. This T cell receptor right here is made randomly using the molecular tricks we talked about yesterday. can it, when born, recognize self-MHC and self-peptide? Absolutely. The T cell receptor has no brain. It was a bunch of DNA thrown together, and that bunch of DNA thrown together throw in a bunch of amino acids. And there's different amino acids. Now, instead of, when I was like this yesterday was with the antibody, now the T cell receptor is really only one arm. Remember, there's a light chain. Yesterday, antibody, the trunk, heavy chain and light chain, and then here's the binding domain. Here's the second heavy chain and light chain. If you cut my arm off right here at the shoulder, there's the T cell receptor. One chain, heavy chain, called alpha. Called beta. Light chain alpha, right there. Stick this in a membrane. We've got a T cell receptor, and every T cell receptor has variable amino acids right here grabbing MHC and peptide. Okay. How did I do all that? Oh. So, when I mentioned that the antigen-presenting cell goes through a draining lymph node and then waits around for its cognate T cell, that's because you only have one T cell that recognizes that one Ebola peptide in that one particular MHC. And that T cell has to cover every single lymph node, and T cells take about 24 hours to go from lymph node to lymph node to lymph node, and they are naive. When they come out of the thymus, T cells are naive. They have not been activated. And their goal, their job when they're naive, is to go from lymph node to lymph node to lymph node, hoping to find its cognate MHC peptide complex, its ligand. If it doesn't find it, it keeps circulating for years. But at some point, when you get influenza, you get mumps or measles, or rubella, or whatever, that T cell, staph, strep, that T cell will make it into a lymph node, and suddenly, after years and years of waiting, find the right MHC with its cognate peptide on that antigen-presenting cell, and now that T cell gets all excited. It finally found its polyglot. Okay. Remember I mentioned that these two cells are adherent, stay adherent with each other for almost 24 hours? So therefore you need adhesion molecules to make sure they don't separate. And what do we need? The T cell, if all we had was this, the T cell hasn't learned anything. The T cell knows that there is a pathogen in the host that I need to respond to. But it doesn't know what that pathogen is, and it doesn't know how to respond. So that 24-hour communication between these two cells is the opportunity for the information that this antigen-presenting cell has gathered when it was in the big toe. It brings that information to the lymph node, and then during that 24 hours, it communicates exactly what are the properties of the pathogen, such that the T cell then knows what instructions it must communicate to the worker bees. Okay. You already know the mechanism of communication. You had a cytokine class, I believe, already, correct? Cytokines are molecules, proteins, made by immune cells and non-immune cells as communication tools between cells. Well, guess what? There's a whole bunch of cytokines that are being made by this antigen-presenting cell. And it's a unique pattern of cytokines made by that antigen-presenting cell due to the fact that the pathogen, the pattern of the pathogen is unique. So the pathogen bound to a unique PRR, pattern recognition receptor. That pattern recognition receptor communicates to the nucleus of the antigen-presenting cell with two messages. One message was make a lot of class 2 so you can present. And the other message is turn on the cytokines that I tell you to turn on, that are important. This antigen-presenting cell makes it into the lymph node. It finds its cognate T cell, and now it's spewing out a subset of the cytokines it's capable of making. And the T cell is getting that information saying, okay, it's a virus. No, it's a parasite. I'm going to need eosinophils, or I'm going to need macrophages, or I'm going to need wherever I need. So these cytokines being made by this antigen cell are unique. The pattern of cytokines are unique. And the uniqueness is established by which pattern recognition receptor was triggered back in the big toe. This unique pattern of cytokines talks to the T cell, which says, okay, I got the info. Let me think this through. Okay, this is what I need to do. Now I will make another unique set of patterns, another unique pattern of cytokines to communicate with everyone else. Any problems? Okay. And since the word is here, polarizing cytokines. When a unique pattern recognition receptor is triggered, it's unique because of the pattern recognition receptor will only recognize patterns of a particular type of pathogen. Therefore, a unique pattern or protocol of cytokines are made by the antigen cell. They're called polarizing cytokines because the T cell that comes out of this 24-hour conversation is polarized to make this group of cytokines and not this group. So these cytokines from the antigen cell talk the T cell into making this cluster and not this cluster. And therefore the T cell is polarized to push an immune response in this direction and not in that direction. That's why they're called polarizing cytokines. They're intel. Think about a general in the Pentagon. The general has spies, and the spies go out and learn stuff. The spies come back to the general and say something, and then the general says, okay, with that threat, I need this and this. Same thing's happening here. This guy is out in the field. He comes back with information, gives that information to the T cell, to the general. It's a CD4 positive T cell, helper T cell. The T cell says, okay, I got the intel. When we strategize, here's my strategy. It communicates to the outside world, to its foot soldiers, what to do to protect us. This is the same picture, a little bit more complicated. And now I'm sort of showing you some polarization. The pathogen causes a unique set of cytokines to be made by the antigen-presenting cell. Those unique set of cytokines talk to the T cell, and then the T cell says, I'm getting remember helper T cells, H for helper, CD4s. I'm going to make interferon gamma. That's the cytokine that's going to keep this host alive. I'm going to make IL-4, 5, and 13 because those are the cytokines that are going to keep this host alive. Or I'm going to make whatever. We have Th1, Th2, Th9, Th7, Th17, Th22. We have a whole bunch of unique T cells that come out of this communication. And each of them are unique by the cytokines they make. T cells have been polarized to make a certain set of cells. Okay? How does this happen? One, the right T cell has to be engaged. It's looking for its cognate MHC and peptide. Cytokines are coming out of the antigen-presenting cell and talking to the T cell. And then we've got this other molecule here. And what is it doing? That molecule is a co-stimulatory molecule. Now, I need to take a side trip again. We talked about the fact that any T cell that sees a peptide itself should be killed because you don't want T cells responding in healthy individuals to healthy tissue. So every T cell that responds to an outside peptide, to endogenous peptide, will be killed off. But we know that's not true. We know there's autoimmunity in the world. So we know that the killing off of T cells that recognize self is good, but not perfect. So we can't eliminate every T cell for every endogenous peptide, it doesn't work. So we always leave some T cells that are armed and dangerous, if you will. So what we add to the system is this co-stimulatory molecule. If a T cell, C cell, in a healthy individual, runs into MHC plus its cognate peptide, its endogenous cell cognate peptide, if you're healthy, that antigen-presenting cell is not going to express CD80. CD80 is only expressed when the antigen-presenting cell is at an inflammatory site. So this is our safety valve. So that self-reactive T cell runs into its cognate peptide, wants to start wreaking havoc, but there is no CD80 on this antigen-presenting cell because there's no inflammation, so it just dies. And we'll talk about that in the slide or so. So if a T cell gets all this information but not this one here, it will die. It was a dangerous T cell. We're in a healthy individual. That T cell got excited. We don't want it to be excited. We want it dead. We want it gone. And the way we do that is requiring it to also see this molecule CD80 in the cell. And the only time an antigen-presenting cell expresses CD80 and 86 is at a site of inflammation. So it's our safety valve that autoreactive T cells will bump into self-proteins, and they will do it in a non-inflammatory setting because we're healthy, and they're pro. And hence, autoimmunity is minimized. Clearly not eliminated because we do have autoimmune disease, but minimized. Good? So that molecule, I think the next slide gives it a name. No, it doesn't. This molecule, this complex, here, you don't have to memorize the numbers. This is called co-stimulation. So right here is stimulating. The only T cell that's going to get angry in the lymph node is a T cell that finds its cognate peptide. That's stimulating a T cell. This is called co-stimulation. It's our backup plan. It's our safety valve to make sure that angry T cells are really only angry in an inflammatory setting. And angry T cells that get angry in a non-inflammatory setting die.
And the inflammation will be from the innate response?
Yeah, exactly. The organisms bind to these pattern recognition receptors and get the whole thing angry. And that's why innate and adaptive are linked. Okay, this polarization of T cells. This is not all the T cells that are out there in this world. There are 10 more. Every day we find more and more subsets. But the bottom line is, if you make this group of cytokines, you get this response. If you make this group of cytokines, this response. You don't have to memorize any of this. It's just an example of that naive CD4 positive T cell in the lymph node found its cognate peptide. It got intel from the antigen presenting cell. That intel from the antigen presenting cell convinced it to make this cluster of cytokines or this cluster or whatever. And then those cytokines in the lymph node will communicate with effector cells, worker bees, to protect it. So, the three parameters of the test question for the adaptive immune response, the three words that will be on the test: diversity, specificity, and memory. We did this many weeks ago, but so I don't have a slide for that. But it's been in red in the various PowerPoints throughout the time. Just want to go over them again. Specificity. That is the fact that a T cell, through its T cell receptor, can only see one itty-bitty 8 amino acid peptide wrapped in the correct MHC. It's the only thing it can recognize. It cannot recognize, you change one of those 8 amino acids, it can't recognize the peptide anymore. You change the MAC from mom's MAC to dad's MAC, can't recognize it anymore. That's the specificity of the adaptive immune response. Antibodies do the same thing. Antibodies can see a peptide of 4 amino acids. You change one of those 4, antibody can't touch it anymore. That's what we mean by specificity of the adaptive response. Everyone got that word comfortable? Good. Diversity. Remember 10 to the 11th antibodies, B cells, each making a unique T cell antibody. 10 to the 11th T cells, each making a unique T cell receptor because we moved the DNA around and we purposely randomized the nucleotides and create amino acids in the binding domain that vary from cell to cell to cell to cell. Every pocket of the T cell receptor, every pocket of the antibody is a little bit different. That little bit of difference, we've got 10 to the 11th differences. That's diversity. We can see everything out there. With 10 to the 11th antibodies, with 10 to the 11th T cells, we'll be able to detect. C is non-scientific. We will be able to detect and therefore respond to every possible molecular signature that exists and or hasn't even been made. And memory is what we're doing now. Just to emphasize diversity a little bit, because I know students trip up on this. When the random DNA rearrangement occurred, this random addition of amino acids occurred, the T cell or the B cell has no idea to which it is, to what it will bind. It's not designing a T cell receptor to bind to everything, every chemical signature out there. It's designing a T cell receptor that doesn't look like its neighbor. And by this randomness, that's how we get diversity. The perfect example, I talked about the Martian, we have antibodies and T cell receptors that recognize Martian peptides or Martian chemicals. And I bet we do. A more concrete example is penicillin. Before we started using penicillin, there are people allergic to penicillin or amoxicillin. Now, amoxicillin was made in a lab. Amoxicillin did not exist until some chemical company, or pharmaceutical company, synthesized a molecule called amoxicillin, or ampicillin, or whatever cyllin you want to talk about. But we have antibodies against each of those. Some of us are allergic to the penicillin family. That's because some of us have an antibody or a T cell receptor whose shape is just right to bind to amoxicillin. Amoxicillin doesn't exist in nature. It only existed after the pharmaceutical company made it. Yet we still have antibodies against it, or T cell receptors against it. That's where the diversity comes in. It's not prescribed. It's not. We don't design T cell receptors. We randomize them, and then if we randomize and make a 10 to the 11th of them, we got everything covered. Now memory. What happens? I don't have a slide for this. Should have. Well, kind of. Yeah, let me go to this slide. What happens in that lymph node? Here's the antigen presenting cell talking to the CD4 cell. Ignore the CD8 for now. Talking to the CD4 cell. They communicate for 24 hours. Here's the MAC and peptide. Here's the T cell receptor. Here's CD80, which is the co-stimulatory molecule. Here are the polarizing cytokines. That T cell becomes angry because we want it to protect us. And it does two things. Differentiation, we talked about becoming these various TH17, TH22, TH2, blah, 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 TH3. We talked about that in a slide or two. The other thing it does is go through clonal expansion. Again, this is a bit of a hyperbole, teaching exercise only. We only have one T cell against the Ebola peptide, and that's the only T cell that's going to keep us alive. But one T cell against a virus, which is making baby viruses every 6 hours, or however long a virus takes, isn't the right ratio. We need millions of T cells. So the other thing that happens is not only does the T cell decide what you unique cytokine pattern should it make, it also makes a whole bunch of siblings. It's a clone T cell. Remember, we only have that one T cell against the Ebola peptide. That one T cell is a single cell. It becomes 2, it's a clone. It becomes 4, it's a clone. This clone becomes more and more and more, and every T cell that comes out of that single T cell is identical to the original T cell. Hence, they are clones, and they have expanded. So in that's another reason the adaptive immune response takes 10 days. You've got to wait till the T cell finds its antigen-presenting cell. You've got to communicate for a day. Then this T cell has to expand. The human cells take about 18 to 24 hours to double. So to go from 1 to 1 million, you need 7 or 8 days to double, to double, to double, and so on. So this clonal expansion and the differentiation, meaning Th1, Th2, Th17, and so on. All that is occurring during those 10 days, and now suddenly you have 1 million T cells that are identical to each other, all making the identical pattern of cytokines to communicate with the effector cell, the working group. Okay. After the organism is gone, we don't need 1 million T cells. Imagine how many infections you get a year. If those 1 million T cells, and remember it's 1 million T cells per peptide, and the organism has 500,000 peptides. So we've got lots of T cells responding to that organism during the infection. Each one clonally expands. Now we've got these billion T cells floating around, all clones of each other. The infection is gone. We want to kill off those T cells because a T cell that's angry with nothing to do is going to start chewing up cell. I guarantee it. So after the organism is gone, we've got to kill off those T cells. We kill off, I don't know, 99%, I don't know the number. We kill off almost all of them, but we leave some behind. And the cells we leave behind are the memory cells. That's vaccination. That's why when you had chickenpox as a kid, you'll never get chickenpox again. Assuming the virus doesn't mutate. Which can ask me about why do we get flu every year? The virus is changing every year. And therefore the T cell memory against the flu in 2017 is not, the flu virus in 2017 is not the same flu virus in 2019, so the T cells can eliminate the 2017 virus, but not the 2019 virus. That's why you get flu again. But chickenpox doesn't mutate, hepatitis C doesn't mutate, rhinovirus doesn't mutate, and go on and on. But some do. HIV does mutate, that's why HIV is such a problem. We can't generate vaccines against HIV because it mutates away very quickly. So memory is those few cells left behind. Now what do we do with them? And notice there are some memory cells. So the million cells that clonally expanded to keep you alive are called effector cells. Once the pathogen is gone, they die off, a couple cells remain behind, and they're called memory cells. By the way, just so you know, there's confusion in the literature. This arrow comes from here. Some people think it comes from there. We do not know if the memory cells are a unique path during this clonal expansion, or if the memory cells are leftover effector cells. We still don't have that information solid. And this is 40 years later. Which is very unfortunate. Oh my God. There we go.
The effector cells are the ones that are attacking the virus? Or what was
Well, if they're CD8s, they're attacking the virus. If they're helpers, they're making cytokines that tell other cells to attack the pathogen. They have the effector cells. They all die off in the pathogen. Almost all die off. And you'll notice here, does the effector cell become the memory cell, or is it earlier on? We still don't know. Years later. Decades of this. Okay? we end up creating two types of, so, I just told you we were going to create two types of memory cells from the slide, but let's circle back a minute. Where would you want to put the memory cells after the pathogen is gone?
Maybe you destroy most of them and just keep what you need for next time?
Excuse me?
Maybe you just destroy most of them and keep what you need for next time?
Right, but where would you put those? The few you keep, where would you want them in the body?
Lymphatic system?
Excuse me?
Lymphatic?
Right, so you could put them, I would want to put them in circulation, so that they're hitting, and the lymphatic system is linked to the blood system, so they go from blood vessel to lymph node to lymphatic to the next lymph node, then pulls out of the lymph node back into the blood and circulates around for years and years and years, literally decades. So we certainly want them in circulation, going from lymph node to lymph node, waiting for that next infection. And those cells are called central memory. And they go from lymph node to lymph node, just like a naive cell went from lymph node to lymph. The other place we put them in lower amounts is in the tissue where you got the infection. Remember, most infections are not cuts to the skin. Most infections are nasal or through ports of entry. Well, some virus or some pathogen is probably going to enter through the same port. So we put some memory cells at the port of entry that was involved the first time, assuming that likely that bad guy is going to come in the same way again. If it does, the effector memory cell will handle it. If it comes in through another port of entry, the central memory cell will handle it by finding it in the lymph node. Here they are. Again, they have proteins on their surface, don't worry about it. Here's everything I just said. Here's that immune synapse that I talked about in the very beginning. We stole the word from the neurologist, neuroscientist. MHC, peptide T-cell receptor, that's specificity. Co-stimulation right here, so that we make sure that we tie T-cell activation to inflammation, so we don't get random T-cell activation when you're healthy, which is autoimmunity. A couple molecules, adhesion molecules, to make sure that these cells don't fall apart. The CD8 cell isn't taking in information to instruct the immune system what to do. The CD8 cell is looking for a cell that is virally infected, and if it finds it, the CD8 cell has the capability of putting out lytic enzymes that will kill the virally infected cell. So even though, antigen presentation, the antigen presentation is used for both cells, but I want to emphasize, and I hope I don't confuse you with this. I hope if I confuse you, ignore me and we'll start again. Antigen presentation is finding an antigen that shouldn't be there, marrying to a lymph node and getting the immune system going. That's CD4. Displaying I've got a virus inside me and kill me is called antigen presentation, but conceptually it's not presenting antigen in the sense of getting the immune system going. It's saying, here's a virus inside me, kill me, because we don't want baby viruses. Antigen presentation is used for both terms. Throughout the hour I've been using that term. It's the only term we use. But I want you to appreciate conceptually they're quite different. Logically they're quite different. Did I lose anyone in that? Good. All right. We've been through this. This is just, okay, we're done. This is, and then I'll take questions for me. I'll take questions for all of immunology for a couple minutes. I'll hang around too. It's 10:48, it's perfect time. Remember I mentioned that in the absence of co-stimulation, this T cell dies. I actually lied. It actually becomes energetic. You'll notice it doesn't say T cell is dead. So remember, a T cell that can recognize a peptide when you're healthy is probably autoreactive, because the only peptides in a healthy individual is cell. The only peptides in a healthy individual are cell. So any T cell that can recognize a peptide in a healthy individual has to be an autoreactive T cell that's going to give you autoimmune disease. We don't want it angry, so we don't have this CD80 interaction, which is tied to inflammation. So when that CD80 is missing, or any way that we make it missing, it ends up, I said the T cells die, I lied to you. They actually become energetic, which means dumb. Now, this is something I do not understand. And I'll leave you with my confusion. So the last time you see me is I'm confused. That's supposed to teach me. If you've got an autoreactive T cell, you don't want it ever to kill you, to hurt you, and you find it, kill it. Instead, make it dumb and non-responsive. The system has chosen to make it dumb and non-responsive instead of killing it. I do not understand why, but that is what happens. In the absence of this co-stimulatory molecule, this co-stimulatory interaction, that T cell becomes energetic, and the next time it sees anything that is cognate for it, it does not respond. So we keep it around. I do not understand why. I would have killed it if I were designing the system. But no one has to. Thanks, guys. It's been a pleasure. I'm not hanging around for a couple minutes to chat.